During this Corona crisis, I spent some time on working on the evolution of nucleotide composition in SARS-CoV lineage. So, so this is our yeah, the dreaded Corona. The technical name is severe acute respiratory syndrome coronavirus 2. So this virus is roughly 30 kb long, and it codes for some important genes like the ORF1 and B, 1A and B are code for replication, and we have other proteins like surface protein, membrane, and nucleocapsid protein and organelle proteins. So before I actually go into the actual evolution, I just want to introduce a little bit about nucleotide composition. So most of the genomes that we know are comprised of like 4 nucleotides, A, T, G, and C. So if we compute the proportion of each of this nucleotide, so in this case it is 30% A, 30% T, and 20% G and C. So the similarity of A and T is obvious because if you look at the sequence, wherever you see A in one strand, it's complemented by T in the other strand. So due to this, now this measure has become simplified, and now we can only calculate the G and C. So this is called GC content because the other part, like AT percentage, is just you can subtract from 100. So this GC or GC content is very unique, and it is generally used for comparative genome analysis because they have it has a unique signature for a species or strain. Now, the GC content of the SARS-CoV-2 virus is 37.97. And my main investigation is to understand how did this level of GC content, the 37.9% GC content, evolved in this virus over time. So for this, I conducted some comparative genomic analysis. So for that, I have to trace the lineage of SARS-CoV-2. So for that, I collected different virus genomes and constructed a phylogeny, and then estimated the GC content for each of those members. So now you'll understand looking at this figure. So this shows the bottommost one is SARS-CoV-2, and its closest relative is RATG, which is a bat virus. And the other two are pangolins, and the next relative is bat one. And the other member of this family is the SARS-CoV virus, which caused the outbreak in 2004 in China. And all these groups are called Sabeco virus. And we have other outgroups like Nabeco and Mbeco virus. And we also have the Mebeco virus, which caused the Middle Eastern respiratory syndrome. So now, so the color coding shows the level of GC content. So here the blue is low and red is high. So when I estimated the GC content for all these individuals, and you can see there's a clear pattern. Like from Mebeco virus, the GC content is declining, and it has got the least for our SARS-CoV-2 virus. So to sort of further confirm this, what I did was estimated the divergence from these viruses from SARS-CoV-2. So basically, you calculate the number of nucleotide differences between SARS-CoV and other viruses, and plotted that on x-axis. and the y-axis contains the GC content. So as you can see, there's a like a positive correlation, so highly significant correlation between the divergence and the GC content. So that means, that means the GC content of the virus that are close to SARS-CoV is low compared to the ones that are distantly related. Or in other words, you can say the difference in the GC content between closely related viruses with SARS-CoV is less than the, so the GC difference increases, widens with the divergence between the viruses. So, so the genome, as you can see, the genome is packed with protein coding genes. So because protein coding genes are important, so they're all under high purifying selection. So because of this, naturally the GC content of the genome will also be under selection. So just to remove that, I looked at the nucleotides, especially the nucleotides that present in the third codon position, which are called as synonymous nucleotides. These nucleotides are generally under less or no selection pressure. So in this case, the GC content in this nucleotides will be much less because of they're free of selection. Then I computed the same relationship and observed the same high correlation. But the only difference is, if you compare these two, the first one based on whole genomes, the difference in, although the correlation was very highly significant, and the pattern is also very similar, but the variation is only 3% using the whole genomes. But if you look at the synonymous sites, it's 9%. So this simply says the less difference is because of the RNA selection. Okay, so now let's, when I calculate the GC content for the distant relatives, up to here, I sort of got a pattern. Then I went up and calculated the GC for this outgroup, which is an Embecco virus, that suddenly dropped down. So that deviates from the pattern. So this simply tells us, like, at this point, so this is the common ancestor of all these viruses. So this common ancestor might have got the highest GC content, and over time, this GC content got declined in SARS-CoV-2 lineage. So, next, so what's causing this GC decline? Because usually the GC content is it's driven by the difference in the rate of mutations. So here we have a rate mutations changing GC to AT and in the reverse direction. So this ratio, what we call is like a rho. So if this ratio is similar for different species, then so the GC content will remain the same for all species. But if rho increases over time, then this will result in GC decline because there will be more GCs changing to AT, so the overall GC content will decline. So I checked this by simply calculating the rate ratios, the rho for different genomes, and as you can see, for MERS virus, the ratio is only 1.2, and this rate increases to 1.44 for SARS-CoV virus. So this again shows the increasing rate ratio pattern, which resulted in GC decline. So this further confirms our finding of decline in GC content. So the next question is, so this is what I observed in SARS-CoV-2 virus. So is this pattern is sort of common? Is it present in other viruses as well. So I sort of repeated the same analysis for the earlier SARS-CoV virus that caused outbreak in 2004. I computed the same thing. I collected the relative the genomes that are related to this virus in a similar way, and then calculated the GC content. And this shows there's no pattern. So the GC contents are more or less similar across the different members of the lineage. And I also repeated the correlation between the divergence from SARS-CoV to other viruses and the GC content. And I didn't find any significant relationship, as you can see from this figure. And again, I did the same thing for the Middle Eastern respiratory virus. And again, I couldn't find the pattern. So this pattern appears to be very unique to SARS-CoV-2. So it appears like so what can we like sort of predict from this finding? So we know though these viruses do evolve, and there will be future viruses that derive from SARS-CoV. So in that case, we could predict that those viruses, potentially will have a GC content less than what is observed for SARS-CoV-2, assuming that the same pattern of GC decline will continue in this lineage. So overall, sort of what sort of application or implications of this kind of study. So right now, the importance is to develop vaccines that are effective against many strains. So not only the current strains, but also the future strains that derive from the current strains. So this study sort of helps to predict what sort of the future mutations potentially will be, because we know the future strains may differ from SARS-CoV-2 mostly in mutations that change from GC to AT. So this sort of gives kind of a predictive power, so that we could have this in our mind while developing a vaccine that is effective against this virus as well as the one that's going to be derived from SARS-CoV-2. So just a summary of the study was the GC content declines in SARS-CoV lineage, and this is because of the differential rates between GC to AT and the reverse mutations. And we didn't observe a similar pattern in other lineages of coronaviruses. And the results predicts, future derivatives of SARS-CoV-2 may have a reduced GC content, and this may be useful in developing the vaccines for future derivatives. And so next, I'm interested to investigate, because so far I've done this analysis for comparing this species or subspecies of viruses. So I would be doing similar things in different strains of SARS-CoV-2, because we have like 40, 50,000 genomes available now, and also compare this, whether the similar pattern we could find in population of viruses that are temporarily separated, the virus that were collected in different time points. So I think that's about it. I'm happy to take questions. Thank you, everyone.
Thank you very, very much, Sankar. That's very interesting and a bit different from what you normally do in a way, in terms of species of investigation. I just want to sort of jump in with a question. When you're looking at where the changes, where the reduction in the GC content is it like evenly distributed, or do you find like equivalent of hotspots where some genes have changed more dramatically? And following from that, there's a lot of conspiracy theories whether this is an engineered virus or not. Is your type of study can shed light on that?
No. So in my study, I found the mutations were totally randomly distributed. So there's no pattern. Although there are, we do know there are some hotspots that's they identified using the population data, but they are mostly at the population level, and that is understandable because even in mitochondrial genome we have hotspots. So that bound to happen. But overall, it is not specific to some gene or some specific site. Yeah.
Thank you. Are there any other questions for? Sorry, any other questions for Sankar from anyone? No, if not, Sankar, thank you very much. That's been fascinating. And
Thanks.
if I can ask you now to please stop sharing your screen. And I would like to invite Trong to give the next presentation.
Yes, yeah, good morning, everyone. Let me share the screen.
Thank you.
Wait. Where is it now? Yep. Can you see my screen now? Can people see my screen now?
Yes.
Yeah,
yes.
okay, that's good. Yeah. Okay, I would like to introduce you briefly about my current projection I'm working on during my fellowship program, STEM Plus Business Fellowship. Just briefly introduce about antibacterials natural drug discovery running at USC. So I bring you with a different field back to chemistry, organic chemistry. So before I talking about before I'm talking about my project, I would like to introduce you a little bit about the library for drug discovery. You may know that to start with the drug discovery program, we normally, the first thing we should consider is the screening library. And of course, we can start to build up the library from different sources. We can start from randomly pick up everything and build up the library, or we can base on the knowledge of traditional medicines or ethnomedicines to build up the library. Or sometimes the families, the species that already know about activities and try to build up the library from that families. Or sometimes you can, with the high technology now, you can use computational chemistry to model the structure and then do the screening. But I should notice that when we have the strategy to build up the library wisely, we normally obtain the better results and increase the chance of success. So I introduce you today the library. I think this is a unique library currently was developed by the CEO of the company I'm working now, Qbiotics. So we call this library is ecologic. So what are the differences of ecologics compared with the other library that make this library become unique? So ecologics is a plant library, and it is all the samples was collected from Queensland tropical rainforest. And even though the Queensland tropical rainforest area is just occupied around 0.1% of the whole Australian continent, but it is considered as the global biodiversity hotspot. And the samples was collected by the plant ecologists. The library contains rare taxonomy, and mostly they are native to Australia. To increase the chemical diversity of the library and also reduce the interference when we're doing the assays, we build up the libraries and we extract the plant's library. We split into 4 different extracts with different polarity. And in that ways, we expand the diversity of the library as well, and trying to collect all the metabolites in the samples. Using ecologic library, the common equibiotics got success in the past when they discover the compounds called EBC46, and now currently it becomes a novel canine anti-cancer drugs on the market. This compound was identified from the Fontania bictrospoma, a native species in Australia, and currently is now in clinical phase 2 to treat cancers, human cancers. I assessed this library to do my research to discover new antibiotics. So just briefly, we did the very traditional screening assays, MICs, on some escape pathogens in humans. And end of the day, we've got the results with a hit rate of 46%. And this was very high percent of hit rate we obtained compared with the other screening. Normally, the hit rate for hydrobus screening in the drug discovery around 1 to 10%. And for special assays, maybe it's lower than 1%. Before I jump up to antibacterials, I want to show you how complex of the fruit extract of the plant. You can see on the left-hand side, the chromatogram of the whole extract of one of the plants we screened. And this chromatogram illustrates how complex we can observe. The extract normally contains hundreds to thousands of compounds from major to minor, but the active compounds normally just one or a few compounds in the extract for specific assays, activity assays. So how we identify the active compounds from a mixture? So that is our role as a natural product chemist. So we got a strategy. So this flowchart will give you the general ideas how we identify the compounds and also how we modify the compounds that can increase the potency of the active compounds. So firstly, normally we have to extract the whole metabolites from the plant tissue. Extraction can be done by liquid extraction or sometimes solid phase extraction. And then further step will be isolation and purification. We will do a series of chromatography steps, very fancy like HPLC, or very traditional ways like gravity column chromatography or thin layer chromatography. But end of the day, we should have the pure compounds to do further structural identification. And we have to, in this stage, we have to collect a lot of spectral data from nuclear magnetic resonance, NMR, mass spectrometries, infrared, UV, combine all that data to show the structure. Sometimes with the minor components, we need to approach some semi or total synthesis to reproduce the compounds or modify the compounds to get better activities, improve the potencies of the compounds, or sometimes modify to get better physiochemical properties of the molecules. And the whole process should be controlled and guided by bioactivity assays. So the assay is also very important in drug discovery. This slide will show you one of the case we obtained, and we got very good results from this plant extract. So you will see on the slides that is the nucleomagnetic resonance spectrum of the pure compounds, and actually this is the best active compounds we've got. So interpret the NMR data and combine with the others mass spec and others data, we finally got the full structure, the three-dimensional structures. This is a small molecules, and normally natural products working with the small molecules, not peptide, not protein. And we also identified the others 9 analogs. They also show antibacterial activities, but different potencies. But they are very activities and comparable to the antibiotic chloramphenicol. Further investigation about the structure-activity relationship from those compounds, they're all new, and luckily we found several positions in the molecules that affect significantly the antibacterial activities. And these results was published last year in Journal of Natural Products. The project still currently running, keep going, and we trying to expand to others antibacterial assays targeting bacterial virulent factors. We already completed a screen on anti-motility assays against several Gram-negative bacteria strains and obtained a hit rate of 7%. And if you notice the figure, like this will show you the anti-motilities of these extracts at different concentrations. Another assays we are developing now is anti-biofilms, and also we're targeting on Gram-negative bacteria. And hopefully in the next few weeks, we will start a screening on the anti-biofilm assays. To wrap up my talk, I would like to thank my mentors at USC's Associate Professor Steve Augment in Natural Product Drug Discovery Group, and Dr. Brett Dales, the CEO of the company Qbiotics. We work this project and should thanks to my collaborator, Dr. David Macmillan, to set up the screening and do all the biology stuff for us. QIMR Professor Peter Parsons and Dr. Jackson Clemens, who's running some cytotoxicity assays, confirm the toxicity of the compounds we identify. And I should acknowledge University of Sunshine Coast, of course, Qbiotics, the company, and SIF funding CSIRO to support the funding for my research. Okay, thank you, and I'm happy to take any questions from you.
Thank you very much, Trong, for presenting your work. Are there any questions for Trong? Trong has been kindly and with the encouragement and support from Steve, currently collaborating with a few members of GeneCology and probably outside as well, helping introduce a biodiscovery aspect to our own projects, which has been really fantastic. Trong, you were mentioning, you know, you're talking about 46% hit rate, which is amazing, and then you talk about 7% here for the antibiotics, which also sounds fantastic. When you say that you got libraries from, you know, from different plants from North Queensland, I assume you get how many different plants, and what is the strategy? Say you find that this library contains something that you want. If you can't synthesize it and you've got to go back to the plant, what is your strategy for them being able to go back and find the, you know, the source?
Right, yes. I'm not a person to collect the sample. There's another team at the headquarters of the companies in Yangabara in Cairns. like I said, they're plant ecologists, and whenever they collect the sample, they already have the record, the GBS of that location. They store, normally they collect around half kilogram to a kilograms, and they store in as the fresh samples in the freezer. And they send to us to do the screening, and whenever after extraction and identify if we lack of materials, they can come back that rate, that locations to collect more samples.
Okay, so they're keeping a record of.
Yes, they have the list of the record, the GBS, and the strategy for them to collect the samples is very clever. That's why we call ecologic. So they're based on the ecological knowledge. So like the way they discover EBC46, because they found that some animals, they ignore that fruit. So they're thinking, oh, maybe that fruit contains toxic compounds. That's why their library contain 100 samples, but they separate into sub-library. So for me, I started with antibacterial screening project, and the way they collect the samples, they noticed that plants or that fruit, when they drop down to the ground, it takes longer time for the leaves or the fruit decayed. So that means that fruit or that leaves contain something that can resistant with the other bacteria, and they collect that samples. So they've got a lot of knowledge in there. So that is the kind of primary screenings, primary selection before we do the screening. So that is the way we increase the hit rate.
Okay, so they're adding smart biology.
Yes.
The power of being smart about the biology, which is beautiful. It really...
So that is very unique. I mean, not because normally you can see the build of the libraries normally randomly, but this is they put the knowledge in there, and the way they select the samples, they just focusing on the rare taxonomy and the very native species, because they want to build off the national Australian, the domestic Australian pharmaceuticals based on natural product, Australian natural products. Yes.
Beautiful. That's really fantastic. And I think this is certainly an area of strength, together with the fantastic analytical lab that Steve led the creation of, and is backed fantastically well by Ecobiotics. And with your expertise, I think it's an area that's really going to grow very strongly for GeneCology and the university, and it's great. Thank you.